this video, we are going to study about the ribosome, their structure and function. What is ribosome? Under this topic, we will discuss about the introduction, definition, discovery, structure, as well as function of the ribosome. As we know that ribosomes are small organelles found in each type of cell, both in prokaryotic cell as well as in eukaryotic cell. Or we can say that this is the only organelle present in both prokaryotic cell and eukaryotic cell. They are the only organelle found in prokaryotic cell. They are not membrane bounded. No membrane is present outside the surrounding the ribosomes. Can be defined. Cell organelle consisting of protein and RNA and function as protein synthesizer is known as ribosomes. Or we can also define ribosome as a ribonucleoprotein particle. Ribonucleoprotein particle is called ribosome. A ribosome complex is made up of different ribosomal subunit. Discovered in 1950 to 1955 by a Romanian cell biologist George Pallet. Approach under microscope is a dense granule, can be seen through electron microscope. A ribosome has two main constituent elements, or we can say that chemically the ribosome is composed of protein 25 to 40% and RNA 37 to 62%. The two main subunits are present as a larger subunit and smaller subunit, or we can say that ribosome composed of large subunit and small subunit. Prokaryotic subunit is made up of large, and their largest subunit is 50s and smaller subunit is 30s. So total ribosomal complex is a 70s. While eukaryotic subunit, larger subunit is 60s and smaller subunit is 40s, and total ribosomal complex is of 80s. So eukaryotic cell has almost 78 protein. Ribosome source, for example, in E. coli, the whole ribosome is 70s, while small unit is 30s and large subunit is 50s. Small subunit composed of 21 protein, large subunit composed of 31 protein. While in rat cytoplasm, which is a mammal, their whole ribosome is 80s and their small subunit is 40s and large subunit is 60s. While in small subunit, the protein is 33, while in large subunit, the protein is 49. What is Sedberg? It is the centrifugal unit depending on the density of the object, and in case of cell organelle determining that how quickly they sink to the depth when centrifuge is known as Sedberg. The quantity of ribosome vary depends upon the type of cell. For example, bacteria composed of 20,000, while in yeast cell it may reach to 2 lakhs. So the quantity depends upon the physiological ability of cell to produce protein. Larger subunit consists of two RNA strands. A longer and a shorter strand drape upon each other. Strands are dotted with protein coats. Protein often glue RNA strand in their characteristics of shape, as you can see here in the diagram. In smaller subunit consists of a single RNA strand. It is also covered with a protein coat, but smaller subunit, though smaller than the larger subunit, is quite enormous than the normal protein. You can see in the diagram. The ribosomal subunit. When we centrifuge them, so 30s, 50s, or we can say that complete 70s of prokaryotic ribosomal subunit. Ribosome can be found either dispersed freely in the cytoplasm or attached to the surface of endoplasmic reticulum and convert the endoplasmic reticulum into rough endoplasmic reticulum. Ribosome is also present within the mitochondria as well as in the plastids. So these are the locations. They are present freely in the cytoplasm, attached to the rough endoplasmic reticulum, and they may be either present within the plastids or chloroplast and mitochondria, which is self-replicating organelle. So on the basis of location, ribosomes are divided into two types, pre-ribosome and bounded ribosome. Pre-ribosomes are found freely dispersed in the cytosol. They are involved in the synthesis of protein that work inside the cytosol. They vary in number depending upon the functionality of the cell type and its need to synthesize a protein. Bounded ribosome. These are found attached to the surface of endoplasmic reticulum and making them rough endoplasmic reticulum. The protein produced by the bounded ribosomes are either transported to the outside of the cells or included in the cell membrane. The main function of ribosome is the translation of genetic information encoded in nucleotide bases of DNA into amino acid sequence of a protein. Simply, we can say that the main function of ribosome is a protein synthesis. That's why we can say that ribosome is an organelle, which it is a protein factory. So this is also known as gene expression. The two ribosomal subunit joins with the help of magnesium ion to translate the messenger RNA into protein. So following are the steps of translation: initiation, elongation, and termination. What is initiation? Or we can say that it is the first step of translation. To begin initiation, the small subunit of ribosomal attached to the messenger RNA. Then the small subunit then move along the messenger RNA until it reach the first readable codon. This codon always codes for the amino acid methionine, which is always known as a starting codon. The transfer RNA molecule are the next to enter. These attached molecule of ribose RNA bring the amino acid to the ribosome. And each transfer RNA has a triplet coding sequence that corresponds with the amino acid it carries along. The triplet sequence of the transfer RNA is complementary to the triplet codon sequence in the messenger RNA. You can see here in the diagram that AUG is present in the messenger RNA and UAG is present in the transfer RNA. So they are complementary to each other. And we know that AUG is code for methionine, which is the starting codon. So each triplet codon sequence in the messenger RNA and the transfer RNA correspond to a specific amino acids. After the first transfer RNA move into the place, the large subunit of the ribosome attached to the small subunit. The complete ribosome consisting of two sides, peptidide site is present toward the left side and amino acyl is present toward the right side. So the second step of the protein synthesis or translation is elongation. After the first transfer RNA is attached to the peptidyl side, a second transfer RNA enters the complete ribosome and attached to the complementary messenger RNA codon in the amino acyl site. When the two RNA in place, the amino acid from the peptidyl transfer RNA move and attach to the transfer RNA on the amino acyl site. You can see clear in the diagram. No longer bearing an amino acid, the transfer RNA move the peptidyl site, leave the ribosome. The ribosome then move along the strand of messenger RNA. A new transfer RNA based upon the triplet coding sequence of the messenger RNA fills the new amino acyl site. These are known as the amino acyl site and these are known as the peptidyl site. The peptidyl site, the transfer RNA leave, while the amino acyl site, the transfer RNA enter. Again, the grooving peptide chain of amino acid is transferred from the peptidyl transfer RNA to the amino acid of the transfer RNA in the amino acyl site. The ribosome moves along the messenger RNA again in another charge with its amino acid. Transfer RNA fills the amino acyl site. For example, you can see here, these are the amino acids. They will cut from this transfer RNA and attach to the amino acyl site, and peptide bond is produced between the two amino acids. So the growing peptide chain is again transferred from the peptidyl transfer RNA to the amino acid attached to the transfer RNA in the amino acyl site. This process of peptide synthesis or peptide formation continue as the ribosome move along the messenger RNA and the future protein grow longer. The last step of the protein synthesis is termination. No transfer RNA exists that recognize the triplet sequence in the messenger RNA known as stop codon. Instead, a release vector enters the ribosome. This structure is known as the release vector. The release vector interfere, attach with the peptide elongation and the ribosome move no further. So the peptide chain is released from the transfer RNA and leave the ribosome. With the peptide chain gone, the ribosome dissociates into its individual large and small subunit. For example, these are the polypeptide chain. These are the transfer RNA. These are the messenger RNA and these are the larger as well as the smaller subunit of a ribosome. Protein synthesis is now complete. The peptide chain is ready to act as a protein or be combined with other chain to form longer polypeptide protein. In this diagram, you can see the whole process of protein synthesis. Ribosome associated diseases. During overburning or stress condition of endoplasmic reticulum and thus ribosomes present on them function improperly. This improper function is characterized by improper folding of protein, As proper characteristics, final folding of protein determine their functioning. So improper folding disrupt the normal functioning of protein, causing a number of deadly diseases. Some of diseases are following. For example, diabetes, cystic fibrosis, neurodegenerative disorder, and other conformational disorders. Latest research in ribosomes. According to Alexander Monken, the ribosome is one of the most complex molecular machine in the cell. His discoveries are the ribosome is responsible for activating some antibiotic resisting genes in the presence of certain protein. Under normal conditions, the ribosome's catalytic center can accept any of the 20 natural amino acids which are then added to the growing protein chains. Other Dr. Hayes worked on prokaryotic ribonucleases that regulate gene expression and cell growth. Nicholas and Golia discuss ribosomal profiling. Three scientists have showed how the information on DNA strand is encoded into protein sequence in ribosome. This proved to be a revolution in the field of genetics. Thus, three scientists have shared the Nobel Prize in Chemistry for year 2009. And the information about these three scientists which shared the Nobel Prize in 2009 is this one. If you like the video, please share, subscribe, and watch.